Okay, everyone, if you don't mind, we're going to get going. We have a lot to cover today. And I also want to make sure that you're aware of what's coming up and what you need to be finishing. So we're going to try looking at some other types of behaviors in the next lecture or so, and we'll have half a lecture in the next class to kind of wrap things up. But some of the behaviors that we look at in neuroscience, you kind of think that they're unique to us as humans, but I'm going to try to make the presentation to you that perhaps some of the behaviors that we think are higher order are in fact not unique to humans, that some of the things that we experience are also experienced by animals as well. So I'm going to tell you a little bit about that as we go along. Some of you will know, if you've been checking on Blackboard as well, that there are some updates. Your literature review assignment is going to be extended by 1 day. If you do need extra time, I need to know that you need extra time by the end of this week. So I don't mind if you need 1 or 2 extra days because you have a whole bunch of things going on, but I don't want you to contact me the night before it's due saying that you need an extension. I'd like you to plan out a little bit ahead of time. Let me know if you think you might need it. If you don't need it, that's fine too, but I need to plan with the TAs. We are on a really tight schedule. We don't know if we're going to be able to get you your marks back because you write your exam on the next to last day of exams. We're going to try our best to get marks in, but there's no guarantee. But we need to have a plan to finish, mark, evaluate, and post things in an orderly fashion. So you will have an opt-in online quiz today. Next week's online quiz, there is one more. We're going to do it together in class. Some of those questions will be found directly on your final exam, so it's something that you should be keeping up to date with. And of course, I'm going to next week talk to you about the final exam, give you some sample questions, talk to you about what to expect, and get you as ready as I possibly can for the final exam. And again, we'll probably talk a little bit more about this as we go on. So I'm going to start off by going back and taking a look at multiple sclerosis, the biology of multiple sclerosis that's so incredibly controversial right now. And as some of you pointed out to me after lecture, last week there was a big announcement that at the St. Michael's Hospital that there was a new institute that was devoted to multiple sclerosis. So Toronto, Canada, we're taking this really seriously. Multiple sclerosis is a big issue. And I'm going to take you through some pathways in the brain that actually we know a lot about, but we don't understand all of the behaviors that result. And we don't understand how the brain can actually influence all of the immune system and how it does so in this sort of psychoneuroimmunology. So I'm going to go back to what we sort of talked about at the end of the last lecture. There are some important things. 1 or 2 of the quiz questions actually come up on this slide. So I told you in the last lecture that multiple sclerosis usually begins sort of as a relapsing, remitting form where we get demyelination. We think it's a problem with the immune system, and as the immune system becomes overly active, it starts attacking different parts of the oligodendrocytes that cause the myelination to occur. And as the immune system starts taking apart the oligodendrocytes, we lose that myelination. Therefore, we start losing our ability to process visual information. So we might have parts of your visual system affected, it might affect the motor system, it might affect speech, it might affect swallowing, all of those things, because we're losing myelination. One of the things that has been tried previously is a drug like mitoxantrone. And mitoxantrone is a drug that is standardly used in the treatment of things like multiple sclerosis. And one of the reasons why mitoxantrone is actually really important is that if you can suppress the immune system and make it so that the overly active immune cells are not quite as active, it might be a way to mediate or to modulate the effects. And mitoxantrone is one of those immunomodulators. It reduces the overall levels of the immune system activation. So this is also in a phase 2 clinical trial. It looks very, very successful so far. It's going on to clinical trial phase 3. And one of the other things that was done at the University of Genoa, and again, there's a lot of research not going on just here in Toronto, but in Canada, but also in Europe, as well as in Italy in particular. And one of the things that was really kind of unique, many of you have heard about the potential of
stem cells. So I want to introduce this maybe in a slightly different way than you might have thought. So could we use stem cells to modulate the effect of multiple sclerosis? It's not the way that you're thinking. In the traditional way, you might be thinking, we'll take stem cells, we'll create new oligodendrocytes. Those oligodendrocytes will therefore go in and remyelinate. That's one potential pathway. At the University of Genoa, though, they actually decided that they would take stem cells and replace the bone marrow. They would actually take stem cells that would replace the bone marrow and repopulate the bone marrow so that the immune cells would begin afresh. So it's a fresh start on the immune system. So if there's something wrong with your immune system, in other words, maybe a better way is to just replace the immune system by starting off with a new set of stem cells that will replace the bone marrow cells that normally will form the white blood cells that would be involved in this sort of immune attack. And then these stem cells actually were found to travel to the bone marrow. They produced and reproduced all of the different cells that would normally be found that would be important in the immune response, and these patients actually got better. So this was one of those initial trials, and this was a trial that, again, had some very significant biological underpinnings. So again, you need to know some of the details on this slide as well. So here's the approach. We can take two different approaches. One, suppress the immune system. Makes sense. The immune system is overly active, it's causing autoimmunity, it's causing multiple sclerosis. That's the mitoxantrone, that's the traditional pharmacological approach. The other way is, let's reboot the entire immune system because there's something wrong with the immune system, and let's see if we remove those hyperactive immune cells, if we can start again. So how do we know if this is really successful? And we need a biological outcome from all of this to be able to determine whether or not this is successful. And so one of the things that they decided to do, instead of just asking patients, are you feeling better? Do you feel that you have better sensation, better motor movement, better recovery? They decided that they would monitor these individuals over a 4-year period, and again, this was published in Neurology. And they did MR imaging. So they did what is now known as T2 weighted images. It's just a different way to take a standard MRI and to look at white matter in a slightly different way, but it is an MRI imaging technique. And they wanted to know if I'm getting either mitoxantrone, which is an immunosuppressant, which traditionally is something we might use to treat multiple sclerosis, or if I reboot your immune system by having stem cells introduced, which one is better? Which one will show fewer problems in the white matter through these T2 lesions, which is a form of MRI? And again, they had to use gadolinium, which is a contrast medium, and again, they tracked this over the course of 4 years. Over the course of 4 years, rebooting the immune system was found to be a better approach overall compared to mitoxantrone. So again, a very different approach to something that we would not normally subscribe to in neuroscience. This is where the interface between the immune system and neuroscience is kind of overlapping. We are more and more interested now as neurobiologists in understanding the role of the immune system in the brain, both during development and in neurodegeneration. And again, in the next 10 years, you're going to see a dramatic shift in the way that we approach neuroscience, and this will be one of those things that people are going to be more interested in. So let me use that now as a preface to get into sort of the next phase of what I want to talk to you a little bit about. So the immune system, how it interacts with the brain, very important. Let me go through other things that also will affect the immune system as well. I'm going to talk to you a little bit about this really powerful concept known as the placebo that many of you have studied in other courses, whether it's a pharmacology course or whether it's a course related to cell biology. One of the controls that we use when we have an active drug or an active substance is to use the placebo. And I went through some of the different standards to determine what a good study is, and one of those was to make sure that there was a placebo that was often involved. So let me give you a little bit of a background, which you don't need to know the details of. I'll tell you when you have to start paying a little bit more attention. Many of you will have heard of some of the different phenomena that I'm going to be explaining to you. In the 18th century, there was a healer known as Franz Anton Mesmer. This is where we get the word mesmerism, or if you're mesmerized, this is where it is derived from. And he actually came up with this idea, or this concept, that he could imbue into inanimate objects or into people this new animal magnetic force that he had determined. And he found this force. It was part of nature. It was part of sort of the fifth element, if you want to think of it that way. And one of the things that he could do using this new force of nature, which again was known as animal magnetism, was to show that if I touched an object and I had this animal magnetism, and I touched this object, and then you came by later, you touched the same object, that you would have an emotional response. It was particularly strong emotional responses like crying, you might faint, you might swoon, all of those things by just touching something that I had imbued magically with my animal magnetic force. And so this was his idea. This was sweeping across Europe. And just like we would do now, when you have a new idea or a new proposal and it seems to be really catchy, it might be really interesting, it might be a new cure for cancer, or it might be something that might benefit individuals with depression, and you see this coming out on the news all the time, you would actually commission a study. And this was done by Louis the 15th. I have to apologize, it's not Louis the 14th, but Louis the 15th appointed a royal commission to determine, is this real? He's making all these claims. People are going across Europe saying that there is this new force of nature called animal magnetism. Is this a real thing or is it not? And just as we would do now in the 21st century, we would hire the best scientists to come in to examine, ask questions, and to determine if there's any scientific evidence-based validity to these claims that I can imbue a force of nature into an inanimate object that causes people to have an emotional response by that transmission of that animal magnetic force. So again, at the time, in the 18th century, they called together the all-stars of science, right? So Antoine Lavoisier, who was the discoverer of many chemical substances, including oxygen, and of course Benjamin Franklin, the preeminent scientists from the United States, they were part of this royal commission. They came in, they wanted to ask Antoine Mesmer, can you show to us that these forces actually occur? This is important for one reason. They were the first individuals to come up with the type of study that we now take for granted. This is sort of a double-blind study. And what they decided to do was to test whether or not this was actually a true force of nature or not. So one of the things that they did was they asked Franz Anton Mesmer to go in randomly into a forest and to touch various trees, to imbue them with this animal magnetism. And then they asked volunteers who were part of this study to go in and to touch the same trees or to touch these trees that he had imbued with his animal magnetism. And they wanted to know, are they going to have this response? Can we see this response? Can we observe this response where individuals will start crying as soon as they touch a tree, or swooning and fainting, or having an emotional response? And so they did this experiment. It's a controlled experiment. But there's one other aspect of this controlled experiment that they introduced. This was also double-blinded in many ways. So the individuals that were going in were told that the trees are over here. And they went off and touched these different trees, and lo and behold, they all had these emotional responses. They started crying and shaking and fainting and everything else. And then they were told, oh, by the way, we just told you the wrong trees. The trees that you actually touched were over there. And so this led to this idea of control and to be double-blinded in these types of experiments. And so at the end of this study, what they basically came up with was, this is actually a real response. They are having an emotional response to something, but these effects were due to imagination. They had to imagine that someone was touching something. Now again, this is in the 18th century. Keep this in mind, that your mind, a Jedi mind trick, for example, can actually produce really powerful effects if, for example, you believe that that's going to actually occur. Now, my question to you is that unique to humans? Are we the only species that can imagine something happening to us and therefore have these types of responses to these types of physiological responses to something that we're imagining? So it turns out that for many, many years, people were kind of going back and forth on whether or not this was a real effect. The animal magnetism of drifted away and no longer was really popular. But people knew that the mind could actually influence the physiological responses that occurred. And in 1955, and published in the Journal of the American Medical Association, you can still go back, you can still download it, you can still take a look at the entire paper. One of the individuals that decided to look at this actually showed very strongly, and this is Henry Beecher, that when he used a placebo, an inert substance, especially related to pain, and in a double-blinded situation. No one knew if they were getting an inert sugar pill or a really active painkiller. No one knew that. The patient didn't know, nor did the physician giving that pill. It turned out that he was able to measure a really strong response that he called a placebo. And in 1955, in an experimental setting that was controlled, Henry Beecher was the first person to actually publish a paper showing that if I took an inert substance that had no medicinal properties, it's nothing but sugar, if I took that, I would actually feel less pain. I would physiologically feel less pain. It was an effect due to the imagination. There is no physiological, pharmacological properties associated with it, and yet it's still causing the reduction of pain response. This is not a sham. This is a real effect. This is not like animal magnetism. This is actually a measured response that is actually now causing us to rethink the way that we do clinical trials in drug studies, etc. A number of different follow-up studies showed exactly the same thing. This is a real response. This was published in 1955, and since that time, no one has been able to show that the placebo response is not real. Everyone measures the placebo response, and it is now becoming something that is standard. We know that placebo effects work on a variety of different systems. So placebo effects don't work on everything. It doesn't matter how strong your imagination is. If you have a broken leg, your leg isn't going to heal any better by having a placebo. But we do know that placebos, an inert substance, if we're given this inert substance, actually will work really well on psychiatric disorders. So if you have major depression, or if you have other psychiatric conditions, and you're given a placebo, in many cases it actually works. You feel better, your mood lifts. If you have neurodegenerative conditions, again, surprisingly, for neurodegenerative conditions like Parkinson's, this actually works as well. Nice. Okay. Particularly true for analgesia. We know that if you are given a placebo, that you have a lessened pain response. This is painful. So again, in 1998, there were a number of reports showing that there is a significant placebo effect when compared to antidepressant medication. So if you were looking at 100 individuals, 50 of them were given a placebo and 50 of them were given an antidepressant medication, that the group in the placebo actually benefited. Even though they weren't getting an active substance or an active drug, they actually benefited from getting what they thought was a type of medication. So again, there's an increasing amount of evidence to suggest that all of these things, pain or analgesia, Parkinson's disease in particular, so Parkinson's is really important because it uses one type of neurotransmitter in particular that's affected, dopamine, and you'll understand this in a few moments, as well as other neuropsychiatric disorders that have been shown in the literature to be affected. There's one other thing that I want you to think a little bit about, and this messes up our studies an awful lot. I don't need you to know all of the details here. I do want you to know what HMB is. So HMB is a measurement of the overall population or size effect. And one of the things that is really interesting about this, if we were to look at the HMB and we were to look at the overall effectiveness of an antidepressant medication by the number of sites or the number of trials, or you could replace this x-axis with time. So you could say on the left axis this could be 1980, then this could be 1990, 2000, 2010, etc. What do you see happening to the population size effect? Is it increasing, decreasing, or staying the same? So all of you should be saying that we actually see a decrease, that overall it was really effective here on the left. Most of the population benefited from having this antidepressant medication, but by the year 2010, that actually the overall size effect, the Hedges G, that statistical measurement that we're looking at is going down. And you have to think a little bit about this. What is this being compared to? So if we're looking at the efficacy of an antidepressant medication, and we're doing this in an evidence-based, double-blinded control, what is our control here? Yes?
Usually in good studies, you would compare it to the best medication on the market.
Okay, so we're looking at the best medication. But what is our control in this study? Placebo. So one possibility is that the drug itself, over time, is becoming less and less active, or we're not able to respond to the drug less or as much as we did before. That's not true. What ends up happening is a phenomenon that we're still not really sure we understand well. The placebo effect in the population is actually going up. So it looks like the efficacy of the drug is decreasing, but the reality is more and more individuals are susceptible to having the placebo effect. So when you're comparing it to a control now, you actually have to account for this placebo effect, which is why this population statistic measurement is important. Yes, you have a question?
Yeah, so just to clarify, like one person taking the drug doesn't feel like there's less of an effect.
No, this is a population study, which is why we're using HMB300. So it's looking at an entire population and how that population is responding over time when we're comparing an antidepressant medication to a placebo. The placebo effect is actually increasing. Okay, so I don't need you to memorize the statistics. A lot of you are kind of looking at that going, that sounds crazy, but it has been replicated that this Hedges-G effect, this population effect, we are more susceptible to placebos than we were many decades ago. I need you to know that Hedges-G is the statistical measurement of a population, not of individual trials. And that's really all I want you to take away. My goal is to talk you through this placebo and this interesting phenomenon known as the placebo. So again, a number of different studies were done, and you can go through the entire timeline. You don't need to worry about these slides at all. And all of these things show that there's an overall, the amount of responsiveness that we see, which is changing, is actually due to an increase in the placebo effect. And when you're looking at large studies and meta-analysis studies using all sorts of different types of drugs, in the most case, placebo is actually doing just as well as the active drug in many of these different cases. So again, I don't want you to get the wrong message from this. I think that the best message I can give you is we don't understand everything about how these antidepressant medications truly work. I gave you some theories about that earlier on in the semester. And then we also have to take into account this placebo effect. There's only a marginal difference when individuals were examined in the placebo versus the actual antidepressant medication trial. But I don't want you to get the wrong impression that these drugs actually don't have a powerful effect on your neurochemistry. I told you that if you were taking these antidepressant medications, or any medication that is affecting your neurotransmitter, that you see these effects almost immediately. We don't get better from depression immediately, but you do see a change in the neurotransmitter levels. So I want you to understand that these drugs actually do have a real effect. It is not just placebo. They have a deleterious effect, especially in younger individuals. So it sounds very contraindicated, but one of the things that is now a black box warning on most of these different types of antidepressant medications is counterintuitive. If you are a young individual, you actually have an increased risk of suicidality and suicide ideation after taking antidepressant medications. This was sort of buried in the data for a very long period of time, and these black box warnings are now part and parcel of these antidepressant medications. The only point that I'm trying to make here is that, yes, antidepressant medications have a very, very strong effect on your neurochemistry, sometimes to a very deleterious effect. And that's not the thing that I really want to get across. It's that when we're looking at the effectiveness of an antidepressant medication, the placebos have a very, very strong effect in addition to the antidepressant medication as well. So remember, placebos work on different types of disorders really well. They work on Parkinson's, they work on analgesia, and they work in psychiatric conditions. So I want you to keep all of that in mind as we're going through some of the other data. So one of the things that if we were to go back to a few slides that I mentioned to you earlier, when Benjamin Franklin and when Antoine Lavoisier were kind of looking at this animal magnetism, one of the things that they said was, it's actually required imagination. And in some ways, we still do need this imaginative aspect of the placebo. We have to think that it's actually going to be beneficial to us. So there are a lot of different factors that underlie this placebo effect. And I'm going to try to convince you that there are some psychosocial contexts. You actually have to think that it's going to help you. There are certain cues that you think will help you that provide enough context that drive the neural circuitry to allow you to experience this placebo effect. There's some expectancy that if someone is standing there giving you a pill as a pharmacist or as a doctor, that placebo effect is much stronger than if you just found a pill randomly and you were to take it. So I'm going to try to convince you of that there's some aspects that involve circuits that underlie behavioral conditioning, so dopaminergic circuits, for example, as well as other higher order circuits that mediate this placebo effect. One of the ways that we can do this is to actually test for what is known as expectation. And I don't know if you've ever gone to the hospital, if you were ever in a hospital recently and you ever needed to have analgesics delivered to you, you know that for the most part now, these are controlled by you. They're patient-controlled devices. You press a little button, it releases a small amount of an analgesic into your IV, and that makes you feel better. But here's the thing. Do you actually feel better if the doctor is giving that to you or if a machine is giving that to you? And one of the things that we can do is to test for this placebo effect and the expectancy of the placebo effect. So again, if you were in the hospital and you were to take an individual who is getting this hidden application, so they're given the exact same amount of drug, and they are given that drug in a way that they don't know they're being given this drug, and then they're asked, how do you feel? Do you feel more pain or do you feel less pain? Versus an individual who has an open application, a doctor or some other healthcare professional comes up to them, how are you feeling today? Do you want something for your pain? And then gives them the open application, or gives them that drug that they see being given to them. Which do you think will actually make you feel better? And again, if you were to use what is known as a visual analog scale, and this is what the VAS is, you are asked to rank your pain. How much pain are you feeling today? So on a scale of 0, no pain, to 10, like the most intense pain I've ever felt in my entire life, what is your pain like? So you might just draw this on this line. They will go and measure this line and measure the distance, and you get an idea of, okay, this is how much pain you have right now. You might then be given the analgesic, and after the analgesic, after it's worked for a little while, the doctor might come over and ask, how are you feeling now in terms of your pain? And you might indicate, yeah, it's a little better, the pain's a little less, so I probably score it down here. And so one of the things that we can do is by using these types of measures is to see, is there actually a reduction in this visual analog pain score, which is what is being shown here. These are some very common types of opioid analgesics, things like morphine. Again, the name's not being important. The fact that they work on the opioid system is important. Morphine or tramadol, etc. And even things that are non-opioidergic, things like ketorolac, all of these, when you take a look, if they're given in an open setting, a doctor is coming in and giving you the medication, the amount of pain reduction that you get in this visual analog scale is always better than if you're given the exact same dose by a hidden machine. So in each case, there's a bigger reduction in pain by about 2 points versus just 1 point for all of these different drugs that we take a look at. We always feel better when we have this expectancy in this open application that we're going to get this medication from a healthcare professional. We are using our imaginative circuits in the brain to help make the pain a little bit less likely to affect us. So is there a physiological and pharmacological response? Yes, because the drugs are actually working. It is reducing pain. But we get a better, bigger effect if we also have the expectancy on top of it. So there are both pharmacological mediators and now more complicatedly, some of these psychological mediators as well. So how does all of this work? I'm going to concentrate on the structures on the right. I want you to be aware, especially as we're talking about Parkinson's disease, that some of these different neural structures that are involved in the placebo response actually involve the dopaminergic system. We know that some parts of the PTA to nucleus accumbens, the dopaminergic parts of the system, will actually help to encode or make the placebo effect much more stringent. If we were to block dopaminergic transmission, for some things, like Parkinson's in particular, it would be effective. But I'm going to be talking to you about this opioid-ergic pathway, the ones that's involved in pain, the one that we know the most about, the one that we can actually map out, and that we can interfere with. And I want to go through this in a little bit of detail with you. We know that this opioid system is part of that descending pain pathway. Do you remember your physiology lectures from a few years back where you were learning? Not at all. Okay, so we know that there are ascending pathways. We can block pain from getting up into the nervous system. So we know we can block that at the level of the spinal cord, local anesthetics, for example, or we can actually increase the inhibition through this descending pathway. We can block pain signals by using these descending signals. And these all involve the neuromodulator known as opioids. We know that this begins in the dorsolateral prefrontal cortex, an area that we're really spending a lot of time on today. And this can also inhibit pain signals by directly activating all of these downstream opioid signaling pathways. All of these are releasing opioid, all of them involve the opioid system. The periaqueductal gray, which is part of the pain pathway. Again, we can also signal to the hypothalamus and cause other types of homeostatic events, but I'm going to concentrate on this vertical line that's part of the opioid system. And we know that we can also activate the rostroventral medulla, or the RVM, and that we can also directly impact the pain signals that we are experiencing peripherally at the level of the spinal cord. We know that this involves the opioid system. Pain can be blocked by endogenous opioids, for example. And we also know that these placebo effects, making you feel a little bit better after having that open application, a physician coming in and giving you those drugs, making you feel better than if you were just given the hidden application, we can take all of that placebo effect away by first giving you an opioid receptor antagonist. Blocking the opioid system at any of these different pathways down here can actually block the placebo effect. You won't feel the placebo effect anymore if you were first given naltrexone or naloxone. Both of those are opioid antagonists. So we know that there's a mechanism. We know it involves the opioid system. We know that these different structures are involved in this descending pathway that involve the opioid. We also know that there are places within this pathway that can interfere with this. Cholecystokinin and the cholecystokinin-ergic neurons can also block these effects as well. So if we were to stimulate and have release of cholecystokinin, we can also block the downstream opioid-mediated placebo effects as well. So these are all important considerations in this placebo. So is there a circuit? Yes. It involves you knowing and imagining that this is going to help. Started off in the dorsolateral prefrontal cortex. It then descends down to the periaqueductal gray, which is a strong place where we get the input of a number of different pain signals. And all of these different pathways can be blocked by the opioid receptor antagonist. So these brain structures are ones that I do want you to make sure that you know. You'll need to know it for your quiz this evening. You'll need to know the actions of different drugs like naloxone, naltrexone, as opioid receptor antagonists. And so I'd like you to make sure that you are aware of this, as well as cholecystokinin or CCK. Nancy, did you have a question?
Yeah, so when you block the opioid pathway, does that increase pain?
It removes the placebo effect. It doesn't necessarily increase pain. There is a slight difference. I know what you're asking, like would you feel more pain if you were to block opioid receptor? It really depends. If you are looking at having an opioid analgesic being used as primary treatment for your pain sensation, blocking those receptors would actually heighten the level of pain very directly, but I'm talking about the removal of the placebo. It gets complicated, as you can imagine. Okay, so let me take you through a couple of things that I do need you to know one of these questions that you'll get as, I think, multiple choice question 5 involves this particular slide. And I need you to understand that we actually have the genetics for this. And you might be wondering, like, why do people care about the genetics? Imagine I'm a drug company. I'm really worried about the size of the placebo effect. I need to eliminate all those really powerful placebo responders. They're going to mess up my data when I'm comparing it to the actual population. And so I need to know, and I need to be able to genotype and figure out what elements of the genome actually allow some people to be better placebo responders than others. And I know some of you are really good at memorizing and really like memorizing. If you do, the next few slides are going to make your day. And if you really hate memorizing, then you're going to have to spend a little time over the next few slides going over some of this data. And so we know now that there are many, many different SNPs, or single nucleotide polymorphisms. And hopefully you remember some of your introductory genetics. You don't need to know a lot, but knowing that you have one single amino acid change can affect the overall function of a protein, including ones that I'm going to be talking to you about, that can dramatically affect your responsiveness to placebo. Many of these involve the serotonin pathway, and all of the bullet points that are here, the 3 that follow under this genetic variant, all of these involve the serotonergic pathway. And one of the things that we know is that we have variations in this serotonin pathway. The first part of this pathway involves tryptophan hydroxylase 2. And in tryptophan hydroxylase 2, or abbreviated as TPH2, in the gene promoter region, we have a polymorphism at position 703. The amino acid at position 703 of this gene, of this particular hydroxylase, means that we can either have a glycine or a threonine there. And this variation actually is really important. If you are homozygous and you have 2 copies, 2 alleles that actually encode for the G variant, you are going to respond better to placebos than if you have 2 Ts at that particular position. So there is a strong correlation from having this particular single nucleotide polymorphism that makes you a better clinical responder for placebos. There is another change as well, which is also related to serotonin, and the serotonin transport system. If you were to look at the gene and the promoter that actually allows for the production of the serotonin transporter, which is the 5-HTP-LLR, if you have the long variant, there are 2 variants. You can either have a variable nucleotide region inserted in the promoter, making it long, or you can have the short version of the promoter. If you have the long variant of the promoter, or 5-HTP-LLR, that helps in serotonin transport, you are a better placebo responder than if you have the short variant. So here are 2 places where serotonin and the modulation of the serotonin pathway can actually somehow affect the placebo effect. Now, why serotonin? We're not 100% sure because it doesn't involve the opioid pathway directly. It probably modulates the descending pathway that I showed you a little bit earlier, but we do know very clearly if you have these genetic variants or these SNPs that you are going to alter your response to the placebo effect. A couple of other genetic markers that make a little bit more sense. Again, one of the enzymes that's normally used to break down drugs and neurotransmitters is the monoamine oxidase. One of those variants is known as the monoamine oxidase A version of the enzyme. And again, in this enzyme, we can actually have a G to T polymorphism as well. If we were to take a look at how these different polymorphisms work, the ones that actually break down the neurotransmitters, therefore having higher activity. So you have higher activity, you have better breakdown of the neurotransmitters, and therefore you might have lower placebo responses, is when we also have these GG polymorphisms within monoamine oxidase A. Again, another one of these molecules that I'd like you to make sure that you know is this COMT, or catechol O-methyltransferase. Do you remember what COMT is used for? Yes? plus, what does the name suggest? Dopamine is one of the classes, one of a larger class of molecules which are known as at the back. Norepinephrine is the other one. So these are together called catecholamines, right? So these catecholamines are broken down by COMT. And we know that polymorphisms in the COMT that break down catecholamines, you'll need to know that, by the way, for tonight, as well as on the final exam. So if this is new to you, or you've forgotten it, or it's been a while since you've encountered COMT, these break down catecholamines, which are dopamine, epinephrine, and norepinephrine. We know that these polymorphisms that occur at position 158, you can either have a valine or a methionine at position 158. We know that we get a lower level of activity of this COMT if you have these MM alleles. You have two versions of methionine at position 158, which means that you are going to have lower levels of the enzyme activity. That means that you have a better placebo response. You will need to know that for your quiz this evening, both what the catecholamine catechol O-methyltransferase is, what it works on, and the overall change in the placebo responsiveness. So nowadays, drug companies are rapidly trying to screen because we have SNP chips that allow you to very quickly determine who in this class, for example, over the course of about 3 hours, which of you will respond better to placebos by having different SNPs related to these different enzymes involved in neurotransmission. This is for your own information. You won't be tested on this. It's more or less a review. So this is that PPH2, which converts that tryptophan into that 5-HTP, which is the beginning of the process of developing serotonin. And we know we have enzymes like monoamine oxidase, which help to break down different types of neurotransmitter molecules as well. So again, this is just for your own information, or if you need to sort of understand how all of these things are working together, you don't have to worry about memorizing anything on this particular diagram, nor anything that I've just pointed out. It's just there for your own information. So I'm going to take you through one last slide, and then we'll take a break. There will be no movement break today, so if you have questions, I'll be happy to stick around. I told you that this descending opioid-ergic pathway that's so important in placebo actually begins in the dorsolateral prefrontal cortex, where we're going to be spending a lot of time today. So we know that in neurodegenerative conditions, including Alzheimer's disease, many areas of the brain get affected. The temporal lobes get affected, many cholinergic fibers get affected. But one other area that seems to be very vulnerable to loss is the dorsolateral prefrontal cortex. So Benedetti and other individuals wanted to know, okay, we get the fact that there are neural circuits. One of those neural circuits starts off in the dorsolateral prefrontal cortex and helps in the placebo response. Totally get that. But we also know that this place can actually be affected by a number of different conditions, including things like Alzheimer's disease. And so what Benedetti and a number of other individuals decided to do was, let's see if this placebo effect that affects this circuit can actually be altered with a neurodegenerative condition. So we know that in Alzheimer's disease patients, if we were to give them a frontal assessment battery test, so we give them a psychological test and a questionnaire, and we get a certain level of responsiveness from them, and we record that score, one of the things that we know is that as Alzheimer's disease progresses, this frontal assessment battery test, their scores will go down. They're not going to be performing as well. And if you were to go back and you were to examine them 1 year later, their FAB tests would be much less. So if you were to do this test with placebo on this Alzheimer's-like patient in year 1, and they had a pretty decent frontal assessment battery test and also a pretty decent placebo effect. They were actually observing a placebo effect. And then you came back 1 year later, where Alzheimer's disease has been destroying certain parts of their brain, including the dorsolateral prefrontal cortex, and you saw that their the frontal assessment battery test scores were lower, showing that their cognitive functions were declining. You would also show and correlate this with the fact that their placebo effect was much smaller or gone after 1 year. So again, anecdotal evidence, correlational evidence, but pretty convincing evidence to suggest that this lack of this dorsolateral prefrontal cortex as part of this descending pathway actually affected this placebo-driven pathway. And are Alzheimer's disease patients actually forgetting the placebo response? That's the way that Benedetti et al. Presented this back in 2006. There's a lot of evidence since 2006 to suggest that yes, you can actually forget about this response by losing parts of this particular circuit. So I think now is actually a pretty good time to take a break. So we'll take a full 10-minute break today, and then we'll pick things up with other aspects of placebo, and then we'll get into whether or not you think placebo and imagination actually can work in animal models as well.
So we have previously looked at the fact that we can block parts of this descending opioid pathway that is involved in placebo. We know that that's the descending pathway that's also involved in pain modulation. And we've been looking at pharmacological inhibitors like naloxone. So we also now have at our disposal a number of other techniques, things like transcranial magnetic stimulation, or TMS. And as neuroscience students, you're going to increasingly see TMS as a really important adjunct therapy. And if this can work to actually block or stop activity in certain regions of the brain, and again, there's some limitations because you're actually using the magnetic pulses to actually block certain areas, but all the other areas above it are also being affected, so keep that in mind. We might be able to block these pathways using a reversible form of inhibition known as TMS, or transcranial magnetic stimulation. And we know that placebo effects can actually be blocked. So the placebo effect, if I give you an inert sugar pill and I tell you it's going to make you feel better, and you do feel better, if I were to block the dorsolateral prefrontal cortex, I would be able to show that the placebo effect on that analgesia is actually going to be reversed, or you won't feel the effect quite as much as you did before. So there are real pathways. It involves opioids. It involves things that we can pharmacologically manipulate. We know that in Alzheimer's disease, as we lose parts of these systems, that it can also be affected. I've been concentrating primarily on the opioid system. I haven't touched the dopamine system, which is also involved in the placebo effect. If you're interested in it, you can go and research it. I just need you to know that the dopamine beta nucleus accumbens has been implicated in the placebo effect as well. But for the interest of time and for the interest of what I need to talk to you about, it's really these systems that I need to get you to think about. So this gets into this question that I started off with. Do animals have this imagination system? Can they actually be fooled into thinking that something is going to have a placebo? You might make the argument that no, this is uniquely human. We are the only species that can imagine a possible benefit when there is no active substance, when nothing else is actually interfering with analgesia or the pain pathway. So if we were to take a look, how would you actually design an experiment to show that there is indeed a placebo effect? And how do you do a placebo type of experiment in animals? So how would I go about looking at naloxone, and how would I show that it's a pain response? And how do I actually prove to all of you that in fact I can fool an animal into thinking that they will feel the benefit of an inert substance in this particular system? So I want you to know the details and how you might set this up in the next series of slides. I don't need you to memorize all of the details of these slides. I don't need you to memorize these tables, the number of individuals that were used in this slide, etc. So one of the most important things that I mentioned to you earlier, in some ways it's almost like behavioral conditioning. If you go to the doctor and you imagine that he's going to help you, it's like operant conditional behavior in Pavlovian conditioning. And so in many ways, we need to look at that same system. But I'm going to try to convince you that this is indeed a way for us to look at the placebo effect. Now, again, one of the things that you can do, you need to have some sort of reward associated with this, and you need some training that's associated with this. You need to know, for example, that if you go to a doctor that you're going to feel better, or that the pharmacist is going to give you a drug that makes you feel better. So we're going to be using a conflict reward assay So they're going to have to believe that a substance that you're giving them is going to allow them to withstand a painful stimulus to get that reward. So there's pain on one hand, there's reward on the other. Is it where they believe that the pain is not going to be quite as bad to be able to get that reward? So we take them through a number of different rounds of training over about 6 sessions. Again, the details not so important. Then they realize that, yes, there's a painful response. A thermal pain is usually about 48 to 50 degrees. Because we don't actually want to hurt these animals, we actually take it only up to 48. They will have to do a couple of things. So in order for them to get a food reward, they have to put their face up against the bar, which then generates heat at about 48 degrees. So they have to learn that, okay, there's a good sweet reward if I go up against the bars, but I also know it's going to be painful. It's going to be at 48 degrees. It's going to be really uncomfortable for me. So again, they learn all of this. That 48 degrees is that painful stimulus. The reward is that sugared water that they would get. So these rats are then set into 2 different groups. They are either given PBS, this is a control substance, or they are given morphine. Morphine deadens the pain response. Morphine attacks or is part of that dopamine system. It blocks the pain going up into the brain. And so these animals that get morphine, they actually don't feel the effects of being up against that 48-degree bar. So they will go up, they don't feel quite as much pain, and they will then take part in that reward. The PBS group, they still feel the pain. Their analgesia hasn't been affected at all. So again, we do this. We take a look. How often do these animals go for the food reward versus responding to the pain? Is that part clear for everyone? So the morphine makes them more likely to get the food reward because they don't feel the pain. The 48 degrees is not quite as bad. If I were to inject, I won't inject you with morphine, but if I were to stimulate the opioid pathway or block certain pathways, the ascending pathway for you, you wouldn't feel as much pain. So you might be able to tolerate a little bit more. And that's what we're doing here. And then we kind of do some interesting things with them. We can continue training them on this PBS, just the vehicle, no effect, versus morphine. It makes them feel less pain, so they will drink more of that sugar water. Even though the 48 degrees is still 48 degrees, we haven't done anything else, they're more likely to take in more of that sugar water. And then we can actually reverse it. We can actually use naloxone. Naloxone is something that we've looked at as blocking the placebo effect. So you need to understand this model. So at this point, if you don't understand the model or you want to ask more questions about the model, it's a good time to maybe ask. Do you have any questions about this or things that you're not 100% sure of? So the 48 degrees painful stimulus, they're going to go up against it. Morphine deadens that response. They've been getting these injections of morphine. Yes?
Why is there 2 conditions?
Condition 1, so there are 2 conditioning tests that they get. So they go through the training twice. So there's always a training phase to this. So you get 2 shots of morphine. You know that, okay, I'm getting a second shot of morphine. I get the first shot, 48 doesn't feel as bad, so I'll get the food reward. I get another shot of morphine. Okay, I got a shot of morphine. I'm going to go up to the 48, and that's that second conditioned test.
Okay, so they just get it twice.
They get it twice. Yes.
And that's enough for them to know that.
I don't know. We're going to get to that in the next slide. Okay, so they get 2 shots of morphine, etc., and then we take them to the next test. So I haven't really shown or demonstrated that this is the placebo effect. So these rats are conditioned to get the response. They get the physiological response. Morphine deadens the response. They are used to getting a shot of morphine, or they get a shot of PBS. PBS doesn't deaden their nervous system responses to the heat pain, and so they are less likely to drink. And this is what you are seeing here. When they get 2 shots of this, they actually don't really see the effect, and they don't drink as much, which is why you're seeing all the clusters down here on the bottom left. And these 2 sessions are enough to get this conditioning done and over with. So when they are getting this, the next time around, so the PBS group I hope makes sense. They still feel the pain. They're not likely to drink. That morphine group, though, that morphine group expects every time I get an injection, that painful stimulus doesn't quite feel as bad, and I'm going to go and drink. So they've gotten 2 shots of morphine. Now you give them, instead of morphine, you give them PBS. Will they believe now that they've been given a shot of morphine, and will they drink more? If they believe that they're getting morphine, they should drink more. That is a placebo effect. And one of the things that you see is in this morphine group that has gotten 2 shots of morphine, and now they're getting a shot of PBS, they in fact do drink more. They actually put their face up against that 48 degree. No, they don't. Yes, they do. They believe that that 3rd shot is going to actually deaden the pain. They are actually drinking more than their counterparts that don't. They are taking part in this placebo effect. Does it involve the same sort of opioid systems that we looked at in humans? So again, it's not all of the animals, about 30 to 40% actually showed this placebo-induced analgesia. And you can also show that you can reverse this effect by giving them naloxone. So does the placebo effect disappear when they're now given, as a 4th trial, this naloxone? Yes, it does. So all of this suggests that in animals, that just like in humans, they also are driven by this placebo-driven effect. They feel less pain, more likely to drink sugared water, put their face up against a painful stimulus, and they believe that when they're not given that morphine, that it's still helpful to them, and that is a pure placebo-driven effect. So I'm hoping to have convinced you that we can study the placebo effect in animals using the same system, that morphine would be the model system versus the saline or PBS control, and that this pain versus reward stimuli are the 2 factors that we need to examine. And all of this is reversible by using that same opioid receptor antagonist, naloxone, that we looked at earlier. So there are some very interesting phenomenon that involves the nervous system. We don't understand all of the mechanisms. We know some parts of the circuit, we know some of the receptors, we know some of the neurotransmitters, but to be honest, we don't understand how all of this works quite yet. And this makes it even more complicated when we start thinking about this system. So there's a lot of evidence to suggest that many parts of the nervous system are responsible for the placebo effect. And I mentioned to some of your colleagues previously that there's also a nocebo effect. Many times you're given an inert substance and you think it's good for you and you do derive a benefit. But if I told you that something was going to be really harmful to you and I gave you this, that this is actually a form of poison and you're going to get cramps after you take it, and it turns out to be the same inert substance, there's also a nocebo effect. Some individuals will also believe that what I give them is in fact harmful, even though it's inert, it's a sugar pill, that they will also derive a detriment to this inert substance. So we know that there's a lot of different things that are going on in the placebo effect. I'm going to try to convince you that there is a lot of connectivity with a number of different parts of the body, and we've looked very briefly at the beginning in terms of the gut-brain axis, but it becomes much more complex when we're looking at what's now known as psychoneuroimmunology. There is actually a lot between the nervous system, the brain, the immune system, and the interplay. So when you think about the question I asked you earlier, do rats have a placebo effect? I'm hoping that most of you will now agree that we can test it, and it looks like it has a placebo effect. And if I were to ask you, do you think that we can induce the same benefits of mindfulness meditation in rats, that I'm hoping to convince you by the end of the next lecture, or the half lecture in the next class, that we can also observe the benefits of mindfulness-like meditation in rats as well, in the same manner. So I need you to understand that this is a complex field. There's a lot of things that I'm not going to spend a lot of time on. This slide is one that you need to know for your upcoming online quiz. We've talked about this previously, and this slide is one that we've looked at very, very frequently. It's one that's kind of misinterpreted by a number of individuals. This is actually showing you homeostasis, and I want you to understand that this is homeostasis. Homeostasis works when we have acute stress. It doesn't really work this way for chronic stress, so keep that in mind. And we know, for example, that stress, psychological stress, will affect your brain. We know that BDNF levels, for example, across the board decrease when you are exposed to stress. That stress response will activate that HPA axis that we've looked at, and overall we will see an increase in corticosterone levels. That's in the short term. Your brain adapts in the short term really well. And when you're looking at BDNF with stress, how is that possible? And one of the things that we now know is that when we have stress, that one of the things that happens in response to stress, that your brain responds to that stress by activating the immune system. And one of the things that happens is that some of the white blood cells will then begin to upregulate a factor known as LFA1, leukocyte factor antigen 1. Again, the long form of the name, not so important here. And what this allows it to do is these activated white blood cells in the short term, in response to stress, will then go up toward the brain. Again, nothing really dramatic there. But in the brain, in the choroid plexus, in response to stress, the choroid plexus will actually increase the amount of what is known as ICAM1. ICAM1 is the ligand for LFA1. So in response to stress, your brain is upregulating this ligand on the choroid plexus, which is part of the beginning of the protective mechanism of the brain. And these white blood cells now are upregulating the expression of LFA1. These bind together and allow the white blood cells to more readily enter into the brain. Why is that important? Because through the entry into the brain, through an upregulation of ICAM1 and LFA1, as a homeostatic mechanism, we've seen a decrease in BDNF. We know that's bad for the brain. In the short term, we get an increase in BDNF because this will now activate microglia to release more BDNF locally. This is a bizarre finding, but it's one that we now know shows a link between the brain, stress, psychology in other words, and the immune system. So this is a well-described mechanism. We also know that there is also a link between psychological stress, which also dysregulates natural killer cells. It decreases their activity and also decreases their production of interferon gamma. Many of you will know that now, around you, that more and more of your colleagues, as you're getting more and more stressed out by all of the literature review assignments, the online quizzes, the final exams coming up, your final assignments and other courses, that you're becoming more and more sick, right? And part of that is because your immune system is being affected by these psychological stressors. And we know that with med students, a prospective study actually showed in med students that there was a decrease in interferon gamma and a decrease in activity of the immune surveillance system cells known as the natural killer or NK cells. So stress students generally have a suppressed immune response. And again, that relaxation will actually reverse that natural killer response cell activity. You don't need to worry about this slide here. There's nothing that's going to be on this slide, but many studies involving med students, dental students, young healthy individuals of your age range actually show that stress dramatically impairs your ability to get better quickly, prolongs illness, etc. So we know that there is a physiological consequence of having stress, not just affecting the brain, but also affecting the immune system. And we need to understand this interplay between all of these different types of interactions. So, it's one of those reasons why actually I want this to be a less stressful environment for all of you. I hope that it has been a less environmentally stressful environment for you, that that if you're coming to class and you are stressed out, you're not going to be a really good learner, and you're going to have a depressed sort of immune system, and that's not beneficial to anyone here. So I want you to start thinking a little bit about this, because I'm going to take you through a couple of studies. Eventually, I'm going to get into the study on mice, and I want to have you understand that this is actually a real phenomenon. It is not sort of pie in the sky, that we don't really understand it, black box, neuroscience shouldn't even bother. It's sort of meditation, mindfulness. It's stuff that belongs in psychology, not in neuroscience. It belongs in neuroscience. There is a real mechanism that we have to be able to understand. So I want you to start thinking, okay, so stress definitely has an effect on the body. I'm hoping that that really brief overview showed you that stress can affect the body. So can we look at other interventions that affect the brain and therefore affect the body in a different way. Not in a bad way, but perhaps in a different way. So we know that 1 month of what is known as integrated mind-body meditation, which is a form of mindfulness meditation, has been shown to reduce anxiety. Now keep that in mind. That's where we're going. In animal studies, we want to show that mindfulness meditation decreases anxiety in a mouse model. We also know that meditation might be working directly by reducing the stress hormones like cortisol. We also know, though, that it's also increasing the activity of other areas of the brain. So we know that the anterior cingulate cortex, an area that we're going to also be spending some time on, is going up in activity. More interestingly, or perhaps more interestingly, we know that the white matter pathways that lead to the anterior cingulate are also increasing. We're getting more myelination over the course of 1 month in the axons that lead to this area known as the anterior cingulate cortex. We can measure this using diffusion tensor imaging, which will follow myelination patterns. So I would like you to start thinking, like, how is it possible that we get a biological effect out of just thinking about meditation or being present in the moment, like just realizing like what's going on around you at any given time? How could that possibly change your brain? So if stress can change your immune response, could meditation not affect your body in exactly the same way? So one of the reasons why I'm introducing this paper to you, there's a couple. One is to introduce a technique that we haven't spent a lot of time on, which is a form of imaging. And another is to introduce this individual here, Michael Posner. So Michael Posner is now retired, but he is still the leader in the field for mindfulness meditation, stress, the effects on the brain, and the overall impact of stress on the brain. So I want you to keep him in mind. You don't need to memorize his name, but you'll see him a lot when we're talking about mindfulness, meditation, the brain, structures in the brain, etc. So we're about to go into the final 2 weeks of class. This is a very stressful time for all of you, and this is actually a time where you're going to have increasing anxiety. And more than any other type of mental health disorder, anxiety is the #1 mental health issue on campus everywhere globally. We need to be able to address this, and many of you will know that mindfulness meditation is one of those things that people have been looking at as an adjunct form of reducing anxiety levels. But I want to understand the biology of this and why people don't really like mindfulness. Part of that is it's a self-report. So do you feel better? Sure. Can I measure that you feel better? Can I measure a physiological response? That's something that most studies up until recently have not done a really good job on. So I'm going to take you through that one paper that I showed you earlier. You don't need to memorize all these slides. Again, I will try to get you to understand what I need you to know. So one of the things that this study was looking at is whether or not smoking behavior can be altered through mindfulness meditation. And there's some really interesting points about this PNAS paper, which is a really high-profile, high-impact journal. So smoking is bad. So I won't lie to you. I don't smoke, but I know that smoking is bad. I've had a lot of friends who've had a lot of health problems, probably due to smoking. It affects every single organ system in the body, and of course it's often described as an elevated or increased risk. And people know this, but they'll continue to smoke. They're somewhat addicted to the nicotine and smoking. They can't stop, even though they know it's a very bad behavior. Again, other sociologists are kind of worried. So those of you who took Soc 101 or an introductory sociology class, one of the reasons why people don't like smoking or having young people smoking, especially teenagers, it's often viewed as a gateway drug. So if you start with smoking, you'll get into other types of drugs. And so this, again, becomes really important, especially in vulnerable teenagers, if they start trying more addictive substances. And this is sort of the beginning of that entry into that phase of their behavior. So there are a lot of different options, but this smoking addiction is still something that affects us. Like it's never surprising to me that one of my colleagues who's in physiology, who knows better, is still outside smoking in between labs, right? And you might see him out there. He kind of looks a little bit like me, and he knows better, but he's actually not able to stop smoking. So some people have suggested, and this is perhaps wrong, that smokers who are persistent smokers just don't seem to have the same level of control as other individuals. And that becomes a real issue. And some people will actually suggest that people who start smoking early, at an early age in their teenage years, they start smoking just because they lack self-control. And so is it possible for us to look at meditation as a way to increase self-control? Is there a structure in the brain that's related to self-control? And can we objectively measure whether or not self-control is actually taking place? Not through a survey about my behavior or how I'm feeling, but actually objectively measure whether or not any of this is actually helping. So again, this is what I do want you to make sure that you know. When people look, and it's not psychologists, it's both psychologists and neuroscientists and neurobiologists, that in some individuals, it's been reported that self-control areas like the dorsolateral prefrontal cortex and the anterior cingulate, there's less activity in those individuals. They're the same ones that are more likely to start smoking or doing other types of addictive behaviors at an earlier age. So these two structures, the dorsolateral prefrontal cortex and the anterior cingulate, are areas that I want you to know have been in the past implicated as being underactivated or decreased in activity in individuals with less self-control. So mindfulness meditation has actually been implicated in improving self-control. So this is where this becomes somewhat more interesting. But there are lots of issues. Most biologists refuse to acknowledge mindfulness meditation, and they kind of push that off to the side. Yeah, you know, we don't like it. There are no controls. There's no biological endpoint. How do I actually know? It's all self-reported. How do we actually know that this is working? Can you show me a structure in the brain that's actually changing after mindfulness meditation over the short term, etc.? So there are a number of issues with this, all going back to that lecture that I talked to you about. What is good science? Why are people having problems with studies that are being published? So this integrative body meditation training, which has been shown to reduce stress, it has been shown to increase attention as well as self-control. And this has been done after a few hours. There are no structural changes, but it's just an anecdotal report. That's not good enough for us. We can't just rely on self-reports. I feel better after a few hours of this mindfulness meditation training. But you could see the advantage. If there is such a big problem with anxiety on campus, and anxiety might be where you simply lack the control to be able to manage your anxiety, how cool would it be after a few hours to have better control over your anxiety levels? And this is why most places on campus are actually looking at meditation and mindfulness as a potential way to get students to be more aware of what is going on. So here's this opportunity for us as biologists now, not psychologists, not sociologists, but as pure biologists to go in and see if this actually works. We need to have an understanding of the controls that we would need to have. How do you control for mindfulness meditation? What is the control? How do I randomize these samples? How do I actually get a biological endpoint that everyone can agree with, not a self-report? So again, these are college-age volunteers, so around your age. So again, it's really good because it reflects the age range of individuals in colleges that have things like anxiety. So we're not looking at anxiety now, but since it's in the same age range, it would be attractive to do so. So again, heavy smokers averaging about 10 cigarettes per day and 33 non-smokers. These were randomized, so that's a bonus. We need randomization. We are not assigning these different individuals to different groups. It's randomized. So one group will go into the integrative body-mind therapy, and the other one will get into relaxation training. So they get 30 minutes of training every single night for 10 consecutive sessions. So it's not a very long time frame. If it works, we should be able to see an effect physiologically as well as in the brain over about the course of 2 weeks. So very, very short. Very attractive. Imagine doing this 2 weeks before your exam started and being able to show improved self-control related to anxiety. So here's where this becomes an objective measure. So they're not asking, like, are you smoking less after getting the integrative mind-body training, or are you smoking less after the relaxation therapy? Again, same number of sessions, same overall idea of relaxing the body versus mindfulness, so very similar looking, but very different in their outcomes. And instead of just asking them, they were actually measuring the levels of nicotine in their bloodstream as parts per million in their blood at various intervals. So they did it at the beginning and then they did it at the very end. And what you're seeing here is that the groups that were randomized, heavy smokers and non-smokers, what you're seeing is that overall, when you're comparing the mindfulness meditation versus the relaxation technique only, that there is a dramatic change over the course of 2 weeks or about 10 days in the integrative body meditation technique. And you can see that the smoking levels, the amount of nicotine found in their blood after 2 weeks or 10 days is much less across the board. For the relaxation therapy group, we actually don't see those changes over the course of 10 days. Interesting. It's a biological outcome. It's not a self-report. They are indeed having less nicotine in their bloodstream overall. And these, again, are heavy smokers and non-smokers. Heavy smokers, you would think that they're addicted to it, and yet we're seeing a decrease there. Another thing that I need you to be aware of is this first bullet point. We need to also identify a brain region that fits with the literature. And when we do a literature review, we know that there are certain brain regions that become important. And one of the things that people do is use what is known as a resting state measure. So at rest, where you're not thinking about something, you're not using your attentional networks, just as a resting state in an fMRI, what we can do is look at what is known as the FALFF, or the fractional amplitude of low frequency fluctuation. We want to know at rest, like what areas of the brain seem to have like overactivation or underactivation. Just at rest where you're not doing anything, are there certain areas? And we know that certain areas for addiction, post-traumatic stress disorder, if you have an attentional deficit, mild cognitive impairment or MCI, and in early onset Alzheimer's disease, using FALFF, we can actually see changes in the overall activity at rest of certain brain regions. If we were to do this, and you were to do this across the entire brain, and you were to do this 2 weeks before they got mindfulness meditation, and then 2 weeks or relaxation therapy, and after doing this for about 2 weeks, one of the things that would be important was that before training, that indeed, like the literature had actually suggested, in heavy smokers, there was actually a reduction in the overall activity in the anterior cingulate cortex and the dorsolateral prefrontal cortex. This is before training had occurred. So did they have less self-control? They're not making that conclusion, but it does fit with the literature about self-control. They have less activation in these areas, especially for the heavy smokers. So 2 weeks after training, there was actually an improvement in the activity. So it was low to begin with, maybe reflecting low self-control, but it was elevated after this IBMT, but not after the relaxation therapy. And again, the areas of the brain being shown here include, for example, the dorsolateral prefrontal cortex. The activity levels do go up after IBMT, but not after the control relaxation therapy. So again, it's an interesting study. It doesn't go through all of the mechanisms. It does show, for example, that maybe it might be useful in self-control. It's definitely helped individuals reduce their cigarette intake over the course of those 10 days, before and after. Doesn't work for relaxation therapy. So it might be really useful for things like addiction, whether it is smoking or perhaps other things like alcohol, opiates, and others. Again, in the past, there's been some caveats. There was no randomization. There was in this study. Michael Posner's study did have randomization. They weren't assigned to these different groups a priori. They were randomized and then allowed to go from there. There was a control in the sense that they also received, the control group had the same length of training and a very similar type of relaxation training, but they saw no clear benefits there as well. The downside is we always want to see a correlation. And one of the things that you would probably be asking, that person there, right at the very top, number 2, in IBMT, they showed a huge decrease. So they're starting off with huge nicotine levels when they were smoking, and after 10 days, they went down to almost zero. So they stopped. Is there an increase? Like, did they actually have the biggest improvement in their dorsolateral prefrontal cortex and anterior cingulate? How many of you would like to know that question or the answer to that question? I think that would be a really important question. But that type of study actually wasn't done. There was no correlation. They actually don't know, and they don't correlate this data, which is a shame, because I think that would be the type of question that any one of us would be interested in. So there is no correlation that they were able to ascribe to for the level of smoking reduction, as well as the overall increase in the FA-LFF in those areas of the brain. Again, there's some mixed literature. So some people say ACC and dorsolateral prefrontal cortex, that's self-control. And some people say it's the posterior cingulate cortex, not the anterior, but the posterior. So the literature tends to be a little bit divided on it. But I think that this is the beginning of what we would want to do as neurobiologists to understand how we can look at and examine this really interesting behavior about self-control, maybe controlling anxiety levels, and being able to manage certain types of psychiatric or psychological conditions. So this idea that maybe this self-awareness, binging as a form of procrastination. So I don't know, are you working on your literature review assignment, or are you thinking about the next installments on Netflix of whatever is really popular on Netflix? And are you going to binge watch that and then try to catch up on your literature review? Do you have that level of self-control? I hope you do. I hope you don't need this sort of integrative body meditation technique, but this would be really good for students to be able to develop. It's one of those things that I asked the provost's office to look into because I thought it was actually really interesting. It's low cost, relatively easy to do. I don't need any specific training, and you might see benefits as early as 2 weeks. And I think it's something that as neurobiologists, biology students, that's something that you should also be thinking about as well. How do you impact this campus as a neurobiology student? Yes, did you have a question? So that's a good question too. Apparently, according to Michael Posner's sort of follow-up interview, not all of them were. So some people didn't want to actually stop, and some people had in the past tried to stop smoking. So that's a good question. So did they want to or not? It's because that would reflect self-control and their inability to control. Great question. Okay, so I'm hoping that when we get to this paper, which is related to meditation training in mice. It's not exactly meditation, but I'll take you through this paper in the next lecture. Can we actually reduce anxiety? So very similar, involving Michael Posner, same ideas, same sort of things that he looked at in humans. This is a paper that came out last year that relates to whether or not there are structural changes that occur in mice and a reduction in anxiety-like behavior with meditative-like brain patterns in mice. And it may surprise you, but the punchline is yes. If you get mice to have rhythmic brain activities that resemble meditation, which you might think is only possible in humans, that indeed they benefit, their anxiety levels go down. So I'm going to take you through in the next few slides what you need to know for the different tests. You're going to need this for your short answer questions on your final exam. You're going to need to demonstrate that you understand how these techniques can be applied and the outcomes of these different techniques. How do we know if a mouse is anxious? And therefore, how do we know if it benefits from having meditation, reducing anxiety? So the one that's the most common is sort of this open field test or this open field activity test. And one of the things that I'd like you to again, make sure that you know on this slide is that if you are looking at this open field, this box that you're seeing, we have software that tracks these mice in real time, and you can actually see how much they're moving. We can actually look at the total distance traveled, the level of activity, if they're hyperactive or more active on the left or less active on the right. All of this tells us a little bit about the level of anxiety. You can also see how often are they standing up on their hind legs. If they're standing up more, they're less anxious than if they're always sort of crouched down or if they're staying in the corners. So for example, you can see that they spent a lot of time here in the corners. And this gets into what this is. What is thigmotaxis? Thigmotaxis. Yes, William.
It's the same as it is in fish. It's that animals like to stay close to walls, which makes them feel
Yep.
more secure.
Exactly that. So if you were to take a look, an animal and a rodent in particular feels much more comfortable when they know what's right beside them. So they like traveling against walls. This is known as thigmotaxis. If they're really anxious, they'll like stay really close to the wall all the time. And this is what you're seeing here. They're really close to the walls. If they're feeling not so anxious, they'll go out and explore a little bit. And this is where they're in the open field. They're also moving around more. All of these are measures in an open field test of anxiety. So if they're moving around all over the place and they're more active, then they're less anxious. If they are sticking near the walls and they're not moving around as much and they're exhibiting sticking near the walls, which we call thigmotaxis, then we know that they're more anxious. And we need to be able to objectively observe whether or not animals are more or less anxious. Another very common test for anxiety in animals is to use an elevated plus maze for anxiety. Many of you in psychology will have heard of the elevated plus maze, a classic in psychology, just as the open field test or the open field assay is. So this is a simple test. They're put on an elevated platform, so it's off the ground. So that part isn't shown here, but they're usually about a meter to about 1.5 meters off the ground. And there are two closed arms where they have glass enclosed walls, and then they have these two open arms. And we want to know if they're feeling anxious, do they like being in an enclosed space, or if they're less anxious, are they in the open space? So the more an anxious mouse spends in the closed arms, or the more time they spend in the closed arms, the more anxious they are. The more time they spend in the open arms, the less anxious they are. We can also see, for example, if they are going into or exploring the open arms more, or if they're exploring the closed arms more. One other aspect of this, if you're taking a look at this slide up here at the top right, one of the things that is really important, at some point, the animal has to make a choice. If an animal is really anxious, part of their brain that's involved in making a choice is the prefrontal cortex. And in some ways, we're not just looking at anxiety-related behaviors, we're looking at decision-making behaviors. This has become a point of controversy in both pharmacology, physiology, neurobiology, and psychology. Is this really a test of decision-making, or is it a test of anxiety? And as a result, the next sort of iteration of this is what is known as the zero maze. The zero maze is also elevated. Therefore, if the animal is looking down and it's anxious because it's so far off the ground, there is no decision-making point. It either stays in a closed part of the circle or it just stays in the open part and kind of looks down if it's feeling really adventurous and less anxious. And this zero maze for anxiety is now becoming one of the standards. There is no decision-making point. It's either against the wall or not. There is no corner that it has to decide to go to or not. And so this is, again, another type of anxiety test that I'd like you to know as a short answer question for the final exam. So another way to tell if an animal is feeling anxious, and this is more for social anxiety, but it can also be used as a general measure of anxiety. Because mice and rodents tend to be fairly social animals, they are put in this type of chamber. On one side, it might be an inanimate object like a Lego piece, and in another, it might be a cage that has another mouse that it's not encountered before. If it's spending more time, for example, with an inanimate object versus socially exploring, this will actually tell you about some social anxiety. A mouse that has social anxiety will actually spend more time with the object than it would with another mouse. So we can use this as another test, and this is a little bit more specific for social anxiety, although some people will use this as a test for generalized anxiety as well. Again, I kind of put the cell phone solution. A lot of you kind of now use your cell phones when you don't want to talk to people. You kind of look down at your cell phone pretending like you're busy doing that. But it is one of those things that we actually want to see interaction, and this is a way to test for anxiety. So another one is the light dark test. We sort of looked at this before. This is actually really hard. It sounds really easy. I can tell you when I was doing graduate studies that this is one of the harder tests to actually get working. So the Morris Water Maze has its own inherent disadvantages, but this one too, because again, it makes more sense. Animals are used to being in the light, in the dark, and if they're feeling really good, they're not anxious, they will actually go and explore in the light conditions. But that's in theory. The reality is you have to really be careful at the level of lighting. So how bright do you actually make it? And it's always one of those things that you have to spend a lot of time titrating, figuring out the right lighting levels, because it doesn't matter if it's too bright. Even if you have an animal who is on all sorts of different tranquilizers, they'll never go into that space at all. So we definitely know that lighting levels are incredibly important, but we can use it if we spend some time on it looking for anxiety. Oops. And the last one, which is here, many of you will already know, this is contextual fear. And anxious rodents actually have greater freezing behavior after a conditioned stimulus than ones that are not anxious to begin with. So we get even more freezing in anxious animals than we do in ones that are just learning about the contextualized fear to begin with. Okay, so I know we've just gone through an awful lot. I'm going to tell you in advance. Some of you are asking, will you have study guides? Yes. You won't have practice questions. As I said, next week in class, we're going to do the opt-in quiz together, and about 2 or 3 of those multiple choice test questions on the opt-in quiz, if you're choosing to do it, will be on your final exam. We're very similar. And the rest of the lecture will actually be a full review. If you can send me any questions related to the different lectures that you're still not sure of, then I'll go through those in the next review session. And again, I'll have everything that you need to study in detail for your final exam. There are some due dates coming up, so please continue to check the announcements page. It's really important. Some of you log in every single day, which is great. Please make sure you're up to date. If you need to see me next week for any of the literature review, please do so. I'm going to be there every day from 11 to 2 o'clock. I'd love to see you. Just make an appointment to come in or talk to your TA. Okay, one last thing. You do know that your literature review has been extended by 1 day to Friday the 8th, I believe. So make sure that you're aware of your requirements there. Let me know if you need any further extensions. Have a great weekend. I'll see you all in the last class next week.